Hello and welcome to this webinar titled New Drug Product Development Using Continuous Manufacturing, hosted by Biopharma Asia magazine and presented by Giustino di Pretoro, Associate Director of Drug Product Development at Johnson & Johnson, and Dr. Robin Meier, Manager of Scientific Operations at Bohle Maschinen. My name is Stephen Edwards and I'll be your moderator. Now please allow me to introduce our first presenter. Giustino di Pretoro is an Associate Director at Janssen Pharmaceuticals, a Johnson & Johnson company. Dr. di Pretoro is a subject matter expert and drug product development lead for continuous manufacturing and coordinator for a series of academic collaborations within the field. Before joining Johnson & Johnson, Giustino was team leader and principal scientist at Novo Nordisk, where he was heading a formulation team focusing on oral delivery of biomolecules, and prior as R&D and business development manager for a Swiss CMO, focusing on novel technologies for particle size reduction and solubility improvement of class 2 BCS drugs. Giustino holds a PhD in pharmaceutical technology and MSc in industrial pharmacy from the University of Milan. Within his PhD, he works as a research scientist at the University of Cambridge in the Chemical Engineering and Biotechnology Department, and later as visiting postdoc in pharmaceutical technology at the University of Düsseldorf. I'll now be handing over to our first presenter. Welcome, Dr. di Pretoro.
Thank you. So welcome everyone to this webinar. We're talking about continuous manufacturing, a technology or platform which I believe is changing, if not disrupting, the way you do development and manufacture of pharmaceuticals. So first of all, I'd like to give a short introduction on what continuous manufacturing is all about, and also share with you some of our experience at Janssen when switching from batch to continuous manufacturing. And then going into a bit more into the topic of the discussion. So what is the impact that continuous manufacturing is having on R&D? What are the challenges and the strategies when developing new drug products using continuous manufacturing? So first of all, what is continuous? Well, continuous manufacturing can actually be different things. We have primary continuous, focusing on drug substance, and here we could further differentiate between small molecules and large molecules. We hear more and more these days talk about continuous bioprocessing, which is gaining a lot of momentum in the industry. And then we have secondary continuous or drug product continuous manufacturing, today mainly focusing on small molecules and oral solid dosages. And that's also the main focus for us at Janssen and the main focus of the discussion today. And then we have the bit more futuristic approach of an end-to-end continuous manufacturing, combining drug substance and drug product. So the approach that MIT and Novartis suggested a few years ago and picked up by another company called Continuous in Boston in collaboration also with the 10 Machinery EMA. Of course, these platforms, they have different goals, but at the end, the purpose of continuous manufacturing is pretty much the same. So that of increasing flexibility, agility, robustness, and quality, of course, of pharmaceutical manufacturing. And then really, modernizing the way we manufacture pharmaceuticals. So why continuous? Why companies like Janssen decided to invest in continuous manufacturing and switch from batch to CM? There are, of course, a lot of advantages. First of all, financial and operational, right? Continuous manufacturing enables a shorter manufacturing cycling time, reduction in footprint. And there is a lot of advantages for the patients, like reduction in drug shortages, accelerated development time, so faster to market for our medicines to reach the patients. And then, of course, quality, because thanks to advanced control strategies and the synergism with PAT, we get to a level of control and understanding of the process, which is way beyond what we can even dream in batch. And this is the main reason why FDA and other regulatory agencies, they are strongly advocating a move into continuous. But I'm sure you have heard this advantages already if you read something about continuous. What you probably haven't seen yet, unless you've seen one of my presentations before or one of my colleagues, is data. So are the advantages that everybody claims on continuous manufacturing real, or is it just speculation? So I'm going to show you some data, at least what continuous manufacturing meant for us at Janssen and what particularly meant for Persista. Persista, for those of you who don't know, is the first legacy product that we at Janssen converted from batch to continuous. And it's actually the first product approved by the FDA as a switch from batch to continuous manufacturing. So for Persista, going from batch to continuous was a huge success. We reduced the manufacturing cycle time from 13 days to manufacture a batch of 1 ton to just 25 hours, which also meant a reduction of approximately 70% in man-hour. We reduced the footprint of more than 50%, the in-process test and release test from 30 days in batch to less than 5 days in continuous. And I believe this is getting even better since earlier this year, FDA approved our surrogate model to predict the solution based on PAT data, enabling a full real-time release testing for Persista. And then again, quality. I mean, what do you do in batch? You take around 10 tablets of a batch of a few 100,000, and you have the presumption that the quality of this 10 tablets is representative for the entire batch. In continuous, we generate a huge amount of data throughout the entire process, feeding into that quality and control that I was talking about earlier. Another thing I want to share with you is, okay, how do health authorities perceive continuous manufacturing? Well, our experience has been very positive overall. We received the first approval for Persista in April 2016 from the FDA, and since then we got approval in 10 additional markets, and more recently last year from Anvisa in Brazil. And this was a huge success for us at Janssen. Is also, I believe, an important milestone in the field of continuous. In fact, when talking to companies who haven't made the switch yet from batch to continuous, their main concern is the way health authorities will, perceive this technology, and not really FDA or even more the emerging countries. Well, our experience has been, again, overall very positive, and it's true that different health authorities that have different understanding in continuous, but they are all very supportive of this technology, and they all see the advantage that continuous is bringing to the patient. Of course, continuous as any new technology comes with challenges. It's more kind of a cultural change compared to the way we've been doing things in batch. One of the biggest change is definitely around control strategies and process understanding. So how do we ensure material traceability in a process that by definition lacks the physical separation of the individual unit ops? And how do we pick up process disturbances and make sure to divert non-conforming materials, etc.? There are also some regulatory considerations, and for instance, we've been dealing internally, so how do we approach, for instance, registration batches or validation batches? Do we follow the same guidelines as we've been doing for batch, or since we generate more data in continuous, we have a better understanding, should we challenge the status quo and do something different? And overall, there's been, kind of a lack of guidelines and an alignment amongst company, and fair enough because it's a new technology. And the situation is changing now with the ICH Q13 being drafted and more pre-competitive collaborations amongst company. Those are very interesting topics, but probably we need a day just to go through them. So in the interest of time, and I focus on the main topic of the talk today, which is the impact that continuous manufacturing is having in development and some challenges and strategies on the development of new drug products using continuous. Well, if it is true that, most of the challenges we are facing in development of new drug products are the same that my colleagues who developed or switched Persista from batch to continuous, we have additional challenges in development. And one of them is the availability of API. API in R&D is quite scarce, and API is quite costly. And there's also variability in API. So the API characteristics at early stage, they are often quite different than API at later stage. And strictly related to this is also material properties and the amenability of these materials into continuous process. So one of the most important aspects of drug product development using continuous manufacturing is material properties. I mean, if I look at my small molecule portfolio, we have more than 30 new molecular entities, and they are all different from each other in terms of material characteristics, in terms of drug load, etc. There is one common thing, though. All these molecules, the majority of these molecules, they are class 2 or class 4 BCS, so they are we're talking about micronized material, generally speaking, fine powders. And we see more and more the need for enabling technologies like spray drying, hot melt extrusion, etc. So the powders we are handling, they are generally speaking very cohesive, they have a very poor flow, and they are difficult to process. Of course, this is not a big difference compared to the situation in batch, but in continuous manufacturing, this might have an even bigger impact. Certain properties like tribalactic charging, for instance, might have a bigger impact in continuous than what they have in batch. In fact, in batch, and even if you have a problematic API in terms of flowability, you will blend it with excipients, and at the end, you will be dealing with the final blend, right? In continuous manufacturing, you have to make sure that your challenging API can be fed through a lossing rate gravimetric feeders, and you might have challenges as those you have seen in the picture. So when it comes to adhesion, bridging, etc. So it's fair enough to say that continuous manufacturing requires a better control and understanding of the material properties compared to what we've been doing in batch. And also important to understand what is the potential impact that these properties might have on the overall process dynamics. And also extremely important is to understand batch-to-batch variability, not only for your API, but also for your excipients. Speaking of material variability, what you see here on the top left is a fixed factor profile. So the amount of material dispensed per screw revolution. For the first NCE we developed in batch. So you see there in the picture 3 different lots of this API. In red is the first batch we basically processed on the line, and then in the meanwhile the crystallization process was changed, so we received from our colleagues from API a material which, according to them, had similar particle size distribution, similar density, similar morphology. And then what you see in blue is an attempt of increasing the flowability, so by removing the fines and optimizing the aspect ratio. So let's forget the blue batch for a while, which is intentionally different. The other 2, they are supposed to be similar, but in reality, they were not, so they behave differently. I apologize, but I couldn't embed a video in the presentation, otherwise you would have been able really to appreciate the differences in terms of flow between these 2 batches. But the one in green definitely had a better flow compared to the others. And what was even more shocking to me was that the batch in blue was, based on common knowledge, like the particles they had a D50 around 50 microns, and they were much more spherical compared to the other batches we evaluated. It actually worse flow than all the others, was the probably most challenging materials we processed on the line. And this got me thinking, so definitely what I'd been underestimating, how complex flow behavior is, and there is not a single descriptor to enable a complete understanding of flow. There is much more than density, particle size, or particle shape, but there is a lot of properties like work of adhesion, cohesion, roughness, nano roughness, etc., that play a major role in flow of your material. And why I'm saying this is because, it's really changed to some extent the way we do development of new drug products in continuous compared to batch. In fact, we have a much stronger and earlier interaction with colleagues from drug substance. So we try to, and also material sciences, so we understand really what are the key properties of the materials we are processing, we're bringing on the line. And together with colleagues from drug substance, we are talking whether we can do any API engineering, so crystal or particle engineering, to make the particles, flowing better and being processed better on the line. So As soon as possible, we perform loss in weight feeder trials, usually offline. And again, we ask colleagues from API to provide as much variability as possible in the materials they give us, so that we can see where there would be or we might be facing any challenges later on in case the material characteristics will change. And speaking of, indeed, material properties, as I mentioned earlier, we're dealing with a lot of, challenging powders in terms of cohesion, pool flow, and variability. And for continuous manufacturing, but then really also just simply for a normal direct compression process, what really matters is that your powder flow and your powder at the end is compressible. For obvious reasons, we are focusing a lot in Janssen on powder flow. And there's a lot can be done here. For instance, via crystal engineering, so changing the surface structure through crystallization. And there are a lot of efforts, not only for us in Janssen, but in other companies, in working on co-processed materials and approaches to make drug substance amenable to continuous processes. And there is also a working group organized by the IQ on this topic. And if you are interested, just let me know, send me an email, I'll put in contact with the organizer. Another approach, which is a bit less challenging than probably crystal engineering, is particle engineering. For instance, by dry surface coating nanoparticles on a host. And one example of this, which we're applying a lot in the company here, is silication. So either co-milling, pre-blending, or pre-treating your API with silica. And what we have seen, and we have applied this to different challenging APIs, is a huge improvement in the flow properties of the material. And again, I'm sorry, I couldn't embed the video because you really appreciate the difference of one of this challenging API prior and after silica has been added. Again, if you contact me, I might be able then to show the video afterwards or to upload it. The advantage, another advantage of silication might also be in reducing variability between batches. I mean, what you're seeing here is, again, a fixed factor profile for different batches of the same API. So even though they are supposed to have a similar size, morphology, and overall characteristics, they do perform differently in a loss in weight feeders. And those are the full lines, basically, you're seeing. As you can see, it's scattered all over the place. Now, what you see in the dotted lines are the same batches after silica has been added. And you notice two things. So first of all, the lines are much closer to each other, and second of all, they are flat. In a simplistic way, the flatter the fixed factor profile is, the better the performance is. Of course, when adding silica, though, you have to consider stability, chemical stability with your API. But you have other, alternatives to silica, for instance, tricalcium phosphate or other excipients that might do as well a good job in improving flow and might be compatible with your API. But definitely, it's an easy and a good strategy. So once we have a good understanding of our material properties and probably we have optimized the API to have it work in a continuous line, the next step is doing really formulation and process development. And most of these development is done offline on individual unit operations. So our strategy at Janssen is to have kind of mirror unit operations. For instance, same type of press, same roller compactor, even the same PET rig and feeders and blenders that we have on the line. And the reason is mainly because the R&D line we have in here in Belgium is dedicated both to development and to clinical manufacturing. So we want to reduce the creepancy of the line as much as we can, and to increase that flexibility and agility in doing the experiments. So we do most of our experiments, for instance, compression profiles, development of static PET models, for instance, for blend uniformity, content uniformity, development of resident time distribution models, all on individual unit operations. And afterwards, we go on an integrated line to perform, additional studies. One of the first studies we perform is an engineering run, a run of usually, 1 or 1.5 hours, where we try to address some of the key aspects when it comes to continuous. For instance, what is the impact of drug load or API, particle size distribution, and some process parameters for line throughput, blender speed, or paddle wheel speed, and again, compression pressure on tablet weight and tablet uniformity, and again, resin mass and time. And after this, we would perform, additional, more extensive DOEs, trying to explore the design space. This step, we also want to understand a bit more about the dynamic phases and the state of control, which are, key aspects when it comes to continuous manufacturing. So we really trying to investigate what is the impact on the overall performance and, for instance, content uniformity during the startup phase. That means, when you are filling up your feeders, your blenders, connection pipes, during your steady state, so when you have an equal inflow and outflow of material, and also during the emptying phase, so when you shut down your feeders, your blenders, and you're emptying the materials basically that is left in the tube. And also in this phase, we might want to see what is the impact of start and stop, and if we leave the line maybe standing over a weekend and we restart the process the Monday after, which is all important aspect when it comes to continuous, which are not or do not really apply to batch development. So after we have a very good understanding then of the process, the next step is control strategy. And when it comes to control strategy, a very important difference compared to batch processes, the use of resin time distribution. And probably some of you on the line never heard this term before, but in a simplistic way, RTD is a probability distribution function, so the amount of time that your material spends into a certain unit operations or in the line, basically. And this RTD is a great tool to, first of all, understand the process dynamics, understand the capability of the line in dampening eventual disturbances, and also track or trace your material. And it's also a great tool that we use in clinical manufacturing to divert non-conforming material. If you want to have more information, just drop me an email, and I will be happy to elaborate this a bit further. But very high level, what we do is that we do determine RTD in the individual unit ops, for instance, the feeders, the blenders, the tablet crest, integrate the line, we validate, verify the model, and then again we use the model as a strategy to divert non-conforming material during clinical manufacturing or eventually during commercial manufacturing. Another great tool then, to control the process, and part of the control strategies is PAT as a standalone in combination with RTD. And some people talk about redundancy, but I think, they are complementary, RTD and PAT. And in addition, PAT might be a great tool to enable real-time release testing, right? So it's something we usually develop in parallel to RTD model. Of course, control comes at a price, right? So especially when developing RTD models, there are hundreds, if not thousands, of samples that have to be taken during development. And that's why interaction and a close interaction with your colleagues from analytical is very crucial. And this might have also an impact on the overall amount of material that you might be needing during your development. And one question that people often ask is, how much material do you really need in continuous? And is it better than batch, or is it worse? Well, the main advantage when it comes to an API consumption compared to batch processes at a later stage, so doing 10 transfers, scale up, assuming that we are transferring from and to a similar line. And if this is, the case, then we'd be saving thousands of kgs of blends. And consider that this stage, our APIs, they're still quite costly, so we're talking about $5,000 to $10,000. It's multi-million saving per compound just from an API perspective, without taking into account other aspects we talked about earlier. Though in early development, not necessarily earlier, we might need a bit more API than what we did in batch, because we have the RTD models, PT models to be developed, and to be fair, we do more in continuous than what we'll be doing in batch. Of course, we want to make the situation even more appealing, because the therapeutic areas, they don't often are willing to invest more material at risk, especially for new products, which still have, a high probability that it would never go to the market, right? So that's why in collaboration with key universities such as Ghent University in Europe and Rutgers in the US, we have been working on API sparing strategies. For instance, development of surrogate models, predictive models, and downscaling approaches to spare material and also to accelerate drug product development. One of the greatest, outcome from this collaboration has been the development of a material property database, so a real library of materials, API, and excipients that offer, in combination with multivariate data analysis, an approach to backtrack, for instance, the effects of material properties on the process, identify surrogate materials, so materials with similar properties to that of your API, but cheap, safe, and easily available that we can use during our development, indeed as a surrogate to the API. And also, they've been very useful in developing this data-driven model, so models linking the material properties to process performance of individual unit operations. I won't go too much into details, but once again, if you are interested, feel free to send me an email. I'll be happy to share some more information. So what we have today is really, we've been developing data-driven models in collaboration with the universities, as I mentioned earlier, for different unit ops. And the next step would be to combine these models into an integrated flow sheet process model. So ideally based on material characteristics and simulating a change in process parameters would be able to predict what is the impact on the quality attributes at the end. Taking a next step, we are also working on interchangeability models. So for instance, model predicting what is the impact if we were to change the throughput of the line, for instance, when we go into commercial, or also if we were to change from a line to another. I mean, we have more and more equipment vendors going into continuous manufacturing, and of course the lines will be different. And we don't want to end up in the same situation as in batch process, where there might be challenges when moving or changing from an equipment to a different one, because then the advantages of continuous kind of get lost, right? So we're working on interchangeability models and de-risking, an eventual change from an equipment or line from a supply A to supply B. And then again, we're working on in vitro models like, surrogate models to predict the solutions using PET data and in vivo models. And this is really the change and the future of drug product development that we are seeing. So really, key take-home message, continuous manufacturing is much more than equipment, as many people think, and it requires an increase in skill set in the organization. Really a big investment, I would say, from pharma companies in finding right people, and so not only formulation scientists, but people with multiple expertise from analytical, material, characterization, PET, etc. But one thing you can be sure, so this initial efforts and investment will pay off in terms of financial and operational advantages, speed to clinic, speed to market, and of course, quality. And of course, none of this would be possible without collaboration. And I realized how important collaboration is actually after I started working in continuous like 3 years ago. And that's because of the interdependency of all these disciplines, as I mentioned earlier. So once again, thank you very much. And if you have any questions or if you'd like to hear more about specific aspects, just feel free to drop me an email at this email address you see here. Thank you.
Thank you, Dr. Pretoro. Once again, I would like to remind our viewers that there will be a live Q&A session at the end of the webinar. So please feel free to send your questions in at any time during the webinar via the questions tab located directly below your webinar screen. I'll go through these at the end of the presentations. So now, please allow me to introduce our second speaker. Dr. Robin Meier is Manager of Scientific Operations at LB Bohle Maschinen, and in his function responsible for the Technology Center of the QB Conline LB Bohle, or scientific collaborations with universities and within scientific consortia. I will now be handing over to our second presenter. Welcome, Dr. Meier.
Yeah, thank you very much, Stephen, for this kind introduction, and also thanks to my colleague Giustino for this great presentation. And yeah, I will be more than happy to follow up on some of the development topics Giustino just mentioned. We'll also fill it up with a little bit of data from different trials, and I will mainly focus on simple R&D possibilities in continuous manufacturing, and to show you how it is possible to move on and to perhaps also to start with continuous manufacturing in your early stage development phases. But at first, I will give you a short introduction on LB Bohle, or let's better say what LB Bohle is doing in terms of continuous manufacturing. When you talk about continuous manufacturing at LB Bohle, you will frequently hear the word QB-Con. QB-Con is the trade name of all of LB Bohle's marketed continuous manufacturing equipment, and is derived from, for sure, the Quality by Design initiative, as you can just guess, and stands for Quality by Continuous Manufacturing. So what we are mainly doing and want to provide is to help you to produce more and higher quality medicines by continuous manufacturing, which is one of the key factors and key benefits of continuous manufacturing, what we just also heard by Giustino. What you see in the background is the technology center of LB Bohle and our headquarters in Ennigerloh in Germany. This is the home and the place where one of our example lines for continuous manufacturing is placed. So here it is possible to test your substances and test your products and formulations together with our experts of our team, and also here seminars are given, for example. And yeah, it is a large showroom to present our thoughts and our visions of continuous manufacturing to you and to interested people. For sure, LB Bohle is not providing and not producing all of the equipment which is necessary to produce complete continuous manufacturing lines. This is also the reason why we need to partner with different other companies on the field of continuous manufacturing. This is on the one hand the industrial partners. We are working closely together with Gericke, a company based in Switzerland, for all continuous feeding and continuous blending technologies, and with the company Korsch from Berlin in Germany for all tablet compression related technologies. Also, we are doing a lot of work in the academic field and a lot of development and research in continuous manufacturing. Mainly, we are working together with the University of Düsseldorf, with the Institute of Pharmaceutics and Biopharmaceutics, with the University of Aachen for advanced control systems and process modeling, and with a new founded open private research consortium, the DDIC, the Drug Delivery Innovation Center, which is a consortium together with different universities, just as the University of Dortmund, University of Düsseldorf, and different pharma companies like Bayer, Merck Germany, UCB, and also Bohle is one of the main partners there. For sure, also we are not the provider for PAT and analytical equipment, and there are a few ideas of companies and technologies we are working together with on our lines we have built so far. And all not only literally meant under the roof of LB Bohle. We are producing and providing all this granulation equipment, all the handling equipment, for sure the continuous coating equipment, and we are the supplier and the developer for the overall control algorithms and the overall steering of the line. Yeah, this is a picture of the QB-Con 25 line. This is just an example line, how such a continuous manufacturing line can look like. We have here two floors. On the first floor, on the top floor, you see the material handling, the feeding, continuous feeding and continuous blending equipment. And on the zero floor, you see all the process equipment. On the left side, you see the continuous granulator and the truly continuous dryer. You see in the middle the tablet press, and on the right side, the sub-batch handling system together with a semi-continuous tablet coater. But just to say, this is just an example how lines can look like. It is also possible to build and to provide lines on 3 or even more floors to have not vacuum transport, but to have more gravimetric transport. There's a lot of flexibility which is possible. And this also brings me to the point that this modular design offers a possibility to do 3 different manufacturing routes and to test them in our test center. This is continuous direct compression, this is continuous dry granulation, together with our own roller compactor dry granulator, or continuous wet granulation when you want to use the whole line. Yeah, but coming to the topic of the talk, the truly continuous drying and the advantages of truly continuous drying. A few years ago, Bohle was also one of the users and the providers of semi-continuous drying. You see here a 3D view on the right side of our old semi-continuous dryer. This means you have had 4 different drying cells and they were drying in parallel, and you created sub-batches after the twin screw granulation step to dry the material. But this comes at a price and with a lot of disadvantages. The first thing is you interrupt the continuous flow, and also you are forced to create sub-batches here which you need to follow up through the whole line in terms of traceability, for example. Another thing, you have highly deviating residence times of your material inside your drying cell, which also means you have highly deviating drying times, because you always have a certain filling time, for example, of 3 or 4 or 5 minutes, and the material which is filled into the bowl, into the drying bowl, is during the filling time all the time fluidized to prevent a caking at the bottom plate, for example, of the dryer. And this means you start the actual drying process, that the material which entered at first into the drying cell is then already almost completely dried. So you over dry this material which enters first. Another thing, setup and instrumentation. It's manifold and highly complex, including the really expensive PAT equipment in this line. And the NIR probes or other probes are susceptible to fouling because they are long time in contact with wet product. And another problem which comes with semi-continuous drying are the high in-process volumes. This means in case of the detection of an out of specification material, you have relatively large amounts of waste you need to discharge. This is the reason why we started to develop and to market a truly continuous fluid bed dryer, which comes also with different advantages, which I will show you during the next slides and which I will underline with data from different trials with different materials. I also want to mention in the right side of your screen, you have the attachment and links there. I added a link, the function, principle, and basic design of the truly continuous granulator and dryer. This is a YouTube video because unfortunately it is not possible to show a video of the function principle here. Perhaps you can run it in parallel, or you can have a look at it later on after this talk, just to get more understanding on how our truly continuous dryer is working. So starting with the setup and the instrumentation, just to give you an explanation what is happening here. The secret is in the simplicity of this machine. It's a linear continuous dryer. You have on the top left side the product inlet from the twin screw granulator, and on the right lower side the product outlet. And then you have a vibrating particle transport grid, so which means the transport functions via a specific vibrator to transport the material from the inlet to the outlet. At the same time, conditioned dried air for the drying of the particles goes through the bottom plate and fluidizes the particles. But it's only a really light fluidization. This is really important. We come later to this point. They are floating that just like on an airbag, all these particles. And then you have an outlet for the drying air, and during the time the material flows through this continuous dryer, it is completely dried. And a few facts coming here. We call it microfluid bed principle. It is possible to integrate the PAT, for example, at the outlet of the dryer, which comes with the advantage that you do not have this possibility to foul the PAT probes via wet product. We have really short MRTs, meaning mean residence times, also a topic we will come to later. And we have nearly plug flow conditions. This is also a specific characteristic of of the residence time distributions. I will underline with data later. And an innovative filter cleaning allows us to run the process for much longer times compared to other filter cleaning mechanisms in fluid bed drying processes. So this is, for example, how the Cubicon 1, the small lab-scale R&D machine, looks like. It's a completely integrated, truly continuous granulation and drying machine and comes together with 3 unit operations. It's the continuous feeding of your powder blend, the continuous twin screw granulation, which happens in this barrel you see just below the powder feeder, and on the lower right side behind this door you have the truly continuous dryer, and all is working via one control panel. This drying principle is also already realized and already in work in production scale. This is a truly continuous dryer for the 25 kilograms per hour line. And what I will show you during these trials are results derived from the following data sets. One data set is a long time stability run for 8 hours, applying continuous granulation and drying on the Cubicon 25. The throughput was 20 kilograms per hour with metformin hydrochloride in more than 90% concentration and around 8% moisture. And the other results are derived from the data set from an experimental plan of the Cubicon 1. This was a trial implementing a placebo mixture with lactose, MCC, and PVP with 2 kilograms per hour and comparably high moisture for twin screw granulation with 25%. And we changed the drying parameters like air temperature, airflow, and transport speed of the dryer. So, starting with the performance of the product filter. Here you see a picture of the product filters of our old semi-continuous fluid bed dryer. This was after a run of around 8 hours, and what you can easily see is that the filters were completely blocked with material. It was a challenging material, but in the end it forced us to stop the trials because we were not able to get more air through the system. And this is one characteristic which you quite often will see in fluid bed drying when you run it for several hours and hours with several sub-batches. And this is one thing we could improve with our truly continuous drying system. You see here on the top left a picture of the filter in the production scale dryer, and now you see also results over 8 hours of experimental run for the differential pressure of the product filter. It's the orange curve, and also the exhaust air fan speed. The exhaust air fan is responsible to maintain a certain degree of underpressure within the drying chamber, which also means when you have an increase of the pressure drop over the product filters, you would also have an increase of the exhaust air fan speed. But what we see here is that over 8 hours, we did not see any significant increase on the one hand of the differential pressure over the product filter, and on the other hand, which is quite logically, when you understand the function principle, we do not see any significant increase of the exhaust air fan after 8 hours. So from the filter point of view, we were able to run this process much longer. But as this is a boring result, we said, okay, let's challenge this filter a little bit more. So we provoked product filter issues. You see again kinetic over time, over a few hours, and on the left side you see the differential pressure over the product filter during the run. It's around 0, so not a large increase. And then we did the following. We decreased the granule moisture in the twin screw granulator. So we decreased the liquid feeding, which means we generate more fines during the process. And this resulted, as you can see, in an increase of the pressure drop over the product filter. Next thing we did, we increased the inlet air volume of the dryer, which also means you do more fluidization during the process. So we see an even more and even faster increase of the pressure drop of the product filters. And we saw, okay, now we slowly run into, let's say, more trouble with the filters. What we did then, we had a look at the filter, and you see the filter is almost completely coated with material. This white material, it's the fines from the granulation. Next thing is, we did a first short filter cleaning pulse, and we do it in a little bit different way than compared to the traditional way of blowback or of shaking or vibrating. I cannot talk too much about this at the moment because of patent issues, but we did a first short filter cleaning pulse, and you see directly that you have a decrease of the pressure drop again. And then we did a second longer filter cleaning pulse, and again we go smoothly back to the original pressure drop of the filter, and we can run the process even longer. So even if we have some problems inside the dryer with the product filters, it is removable over this innovative filter cleaning mechanism. Next thing I also mentioned, the susceptibility to fouling of the NIR probe. What you see here is that we have the NIR probe. This is a magnification on the right side of the outlet of the dryer. We have the NIR probe directly at the outlet of the continuous dryer. So we are able to measure different product characteristics. In a classical way, when you do wet granulation and drying, you measure at this point the loss on drying, or let's say the residual moisture of the granules. You can also detect even more when you're experienced in multivariate data analysis and PAT. You know how powerful PAT probes on this position can be. But what we classically do is measuring the residual moisture of the granules there. And what I mentioned is that you can have problems with fouling of your NIR probe windows when you run your process, for example, semi-continuously, and when your NIR probe is in longer contact with wet product. This does not happen here in this case. After a first phase of controlling of the process, we have at first a short increase of the moisture. We have during the other 7 or 8 hours of the process a really smooth and steady measurement of the LOD of the granules, and we do not see any fouling. If we would see fouling at this point, we would see a very flat curve, so no changes over time anymore. This is usually a characteristic for fouling, because then the NIR probe always sees and measures the same product. We do not see it here, and this also gives us right that this probe position is a really well-designed position to have a long and stable running time also of your NIR probe. Going further to other advantages like residence times or residence time distributions, which at the same time in this specific field means also drying times. Giustino in the further talk mentioned it shortly. Here you see derived from a full factorial DOE, different residence time distributions inside the dryer. What we did, we spiked the throughput of the dryer with colored granules and measured the color distribution at the outlet of the dryer, camera-based. And what you see when you are experienced in residence time distributions, that you have almost symmetrical curves. When you have a large amount of backmixing, for example, you see a long tailing usually at the end of the curve. This you do not see here, and this is what I also meant with nearly plug flow conditions. So you have only little amount of backmixing, which is quite advantageous for this drying process, because you want to dry the particles evenly. You do not want to have large distributions of residual moisture throughout your granules which getting dried. For example, here we created different flow conditions, different inlet air amounts, different traveling, so vibration speeds, and you can really control the drying process quite well with the residence time distributions. And this you can see even better when you normalize these residence time distributions. You can normalize residence time distributions by easily dividing all these numbers you have here by the mean residence time, and what you can see then is when you normalize them, they are almost identical. This means when you scale it to different throughputs or scale it to differently sized drying machines, it's only the mean residence time you have as a factor for the scale up. It's nothing to do with the width of the distribution, the amount of backmixing, and different other characteristics you can pull from the mathematical description of this residence time distributions. It's quite easy. It's only the mean residence time you need to have. And these residence time distributions, which are really sharp and really short, which you also see here, have impact on different things. Giustino also talked about startup and shutdown conditions, time to reach the state of control, for example. The shorter your residence times are, the faster you get into the state of control. And this is one important point and one important take-home message you need to have. Also, the in-process volumes are really low. I will come back to the in-process volume point at the last slide there, where we measured the mass inside the dryer. Then you have only little amount of backmixing here. Backmixing is always the thing you want to have inside, for example, your continuous blenders, but you only want to have as much backmixing as necessary. Because when you have too much backmixing, you have too much smearing of, for example, different sub-batches you create. And this might give you difficulties with regard to the traceability in the end of the process. And another impact is the diversion in case of out-of-specification material through disturbances. When the residence time, for example, is really thin and narrow, you do not have so much backmixing of your OOS material, and then you do not need to divert large amounts of material in the end. Another impact of the residence time distributions is the residual moisture in the different size fraction of the granules. We divided them into different size fractions after the dryer, smaller than 0.5 millimeter, granules from 0.5 to 1 millimeter, from 1 to 1.4 millimeter, and larger than 1.4 millimeter. And you see only a slight difference in the residual moisture in the granules larger than 1.4 millimeter, but here you can discuss if this difference is of any relevance here. I think it is not, but it needs to be mentioned. And what you see is, and the take-home message here is that it's possible to dry material and granules from different sizes evenly throughout the dryer. Next point, and next important point, not only for the raw materials, the particle size distribution is important, also for the dried granules and for the later feeding and tableting process. It is important to have a really narrow residence time distribution on the one hand, and also to generate not too high amounts of fines which can alter your feeding or your tableting process. And what we did, we measured the granule size distribution before and after the drying to see what is the influence of the drying process for the granule size distributions. What you see here is a cumulative granule size distribution. We collected the granules manually after the twin screw granulator on a tray and let it just dry on a tray. And afterwards we did a camera-based particle size distribution analysis. And then we also measured, after full factorial DOE with around 11 runs, we measured the granule size distribution at the end of the dryer. And what you can see, this is quite interesting. I will try to jump back and forth. You can see that the granule size distributions after drying are almost of the same magnitude, and the curve is also of the same shape as just before the drying process, independently from the drying parameters, just as airflow, air temperature, or velocity of the material inside the dryer. This means that you do not destroy your granules in the drying process. It's a really gentle drying process where you do not create excessive amounts of fines. And also we evaluated this via the statistical software. And what you see, usually in this coefficient plot, you do not like to see these results, where you have this statistical bars going over the 0 point. But this shows you that none of the drying parameters or the parameter combinations has any significant influence on the D50 of your material. And this shows and is a proof that the drying process does not significantly influence the particle size distribution. Next point, you have an improved control of the drying process. You see here again different residence time distributions we created inside the dryer. And what we did, we had a constant twin screw granulation process, and measured the residence time distributions and also connected this to the measured loss on drying, so the residual moisture of the granules after the dryer. And what you can see, which is quite logical, is that you get a decrease of the granule residual moisture when the granules are longer inside the continuous dryer, up to a certain point you see on the right side, which is red marked. At this point, the longest residence time distribution, you have again an increase of the residual moisture, and also the variance is higher again. At this point, we reached, and so to speak, this is an unrealistic point. It was just to test the boundaries of the system. But at this point, the powder bed or the granule bed height is too high, and with the same drying parameters, you cannot dry the granules evenly anymore. But this is only the boundary for the vibration speed. When you would reach this point, and when you say, okay, I really want to work on this point, you have still two different possibilities. You can still increase the airflow to have, again, a steady and smooth flow through the particles, or you can also increase the temperature to improve the drying process. And this was also evaluated via a DOE, and you see on the left side the model descriptors, and on the right side the coefficients for the drying air temperature, the airflow, and the vibration. And all these factors are independently able to influence the drying process and the drying results of the granules after the drying process. So, last point I mentioned in the beginning, this is the fact that you do not need to discharge too much material when you have or detect out of specification material. You see in this plot the mass of granules inside the dryer as a function of the mean residence time. What you can guess, when you increase the mean residence time, the material travels slower through the system, and you also have an increase of the mass inside the dryer. But what we see here for the Cubicon 1 is that we have only masses from 15 up to around 70 grams inside the dryer at the same time. And this is a really low number of material. And this is also significantly to reach quickly a steady state. And brings me to the next point on the right side. This is the time we needed for an 11-run design of experiments on this line. And yeah, we started at 9:14 in the morning and ended at 2:30 in the afternoon. We were just interrupted by a lunch break, and the aim was to collect 200 grams of material for each process condition. So in the end, if you say, okay, I perform a DOE and do not need to collect so much material, you can get your DOE even faster. But another key take-home message is you get your DOE, for example, in your research and development facilities done within 1 day or half of 1 day. And you get a lot of data and a lot of knowledge to evaluate and to design your processes even better. So you can say problems of semi-continuous drying were solved so far via the introduction of the truly continuous drying process. And I would be more than happy to talk with you about perhaps questions of the continuous granulation and drying process. Contact us for further ideas and questions. Also contact us when you are interested in visiting our test center and testing with your material on our small or our large lines. Yeah, just feel free to contact me. Thank you very much for the attention.
Thank you, Dr. Meier. Now we will begin the question and answer segment of the webinar, but once again I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question is, what is the strategy in division at Janssen for development of new drug products? Giustino, would you like to answer this?
Sure, Stephen. Well, our strategy is really to use continuous manufacturing as a main platform for drug product development, all the new chemical entities. So we started a few years ago with Presista as a switch from batch to continuous as a legacy product. We continued with another switch from batch to continuous with Tramoset, recently approved by PMDA in Japan. And now the real strategy for at least new products is to use continuous manufacturing as the main or preferred platform in the organization. So there needs to be a strong rationale for let's say, diverting and still, continue using batch.
Thank you. Our next question is, what is the Janssen deployment strategy in continuous, and how many lines are available in the J&J network? Again, Giustino, would you like to answer this?
Sure, Stephen. So we have currently 3 lines in the Janssen organization. So the first line is a line where Presista was developed, and the manufacturer is located in Puerto Rico, Gurabo, in our first supply chain. We have another line focusing on wet granulation in our supply chain in Italy, Latina, and an R&D line here in our R&D center in Beerse, Belgium, which is for development and clinical manufacturing. It's a bit more flexible line, capable of doing direct compression, wet and dry granulation. And the strategy then is to basically invest in a mirror line, what we call, so it's a line with similar capabilities to what we have here in R&D, and this line will be installed in our supply chain in Latina. So the strategy, once again, is to do development in R&D and do launch and commercialization from the other line in Latina. And then we are talking still about the future and investments of other lines. Thank you.
Our next question. How do you ensure a robust measurement with your PAT tools at the outlet of the dryer? Dr. Meier, would you like to answer this?
Yeah, thank you for the question. In this case of the dryer, we have different measures. So we have a rotary valve at the outlet to have a certain product level always covered with the NIR probe. And we also using different kinds of fill level sensors which control the rotary valve. So the aim is that the product is in movement all the time, or also when your PAT strategy is different, that you always only want to measure non-moving powder. You can also proceed this way, but the idea is to keep a constant fill level controlled by the fill level sensors and the rotary valve, so that the NIR probe or the other probe you want to implement there always sees product, that it never measures air or something like this. So that you have a more stable measurement, and that you always have the same density of material in front of your probe window. This is really important to have a robust measurement with the PAT tools.
Thank you. Our next question. What is the preferred technology platform for new product development using continuous manufacturing at Janssen? Giustino, would you like to answer this?
Sure. So we're trying to use direct compression as the preferred platform, and that's because it's cheaper, faster technology compared to wet or dry granulation. The main challenge, as I mentioned earlier, is really the amenability of properties to direct compression, really. So that's why we are collaborating a lot with our colleagues from drug substance in improving the material properties so to enable direct compression preferred platform.
Thank you. Next, we have, how do you measure the residence time distributions of the granules within the dryer? Robin, I believe this is for you.
Yeah, I think so too. So what we did, I just mentioned it briefly during my talk. Usually when we want to do residence time distribution measurements only with a dryer, we produce colored granules. We do it on a visual basis, so it's a camera-based tool. The thing is called ExtroVis. And with this camera-based tool, we spike the throughput with a certain amount of colored granules. Usually we use, for example, red granules. It's just because it's the best way to detect the granules. Strangely, the color red is working best. And we spike it at the inlet, and at the outlet we mounted this inline camera, and with this camera we directly measure the color intensity at the outlet, and thereby we get directly our residence time distribution curves. The software allows also to process it further and so on, but usually we use the raw data and do our own processing, but this is just personal gusto, let's say.
Thank you. Moving on. Are you also able to process micronized material with a comparably powerful product filter performance? Again, I believe this is for you, Robin.
Yeah, thank you. Also a really good and also impressive question. Yes, the answer is yes. We did a lot of research, especially in this field, to also work with micronized material, because we also heard in Giustino's talks that most of the materials, because of the BCS2 or 4 class characteristics, are micronized because of bioavailability reasons. That's the reason why you really need to be able to work with micronized material, and the answer is definitely yes. We improved the filter cleaning even more, and we have special filters and special material to also run these long times with our dryer, also with micronized material, without losing the material and also without blocking the filters after a certain amount of time.
Thank you. Our next question. What percentage of SiO2 is, generally speaking, okay for many APIs? Giustino, would you like to answer this first?
Sure. I don't think there's a big difference between batch and continuous manufacturing. So the silication approach, or the, let's say, amount of silicon dioxide that you would add is pretty much the same as you would do in a conventional batch process. In our experience, we have seen amount as low as 0.1% of silica working very well, and the same consideration as for batch. So you don't want to put too much silica, otherwise you have a counterproductive effect than what you'll be having. So it's not a big news that by adding a lot of silica, then surprisingly your forward flow might get even worse than what was in the beginning. So I would say anything between 0.1% 0.5% might work fine.
And our next question is, what are the typical questions you receive from FDA and EMEA, and what is your impression of other regulatory authorities, specifically Japan, China? Giustino, would you like to answer this?
Sure. So I'm not sure what exactly, the exact questions, especially for persistence, since I was not in, but the impression we have, definitely interacting with different regulatory agencies, is that they have, as I mentioned in my talk, a different understanding. So you have FDA having a huge understanding now of continuous. They've been, collaborating also with universities like Rutgers. They even purchased their own small equipment. So they know a lot about continuous manufacturing today. Well, the situation was different when we, started working on Persista in around 2008, 2010. And there, we were in the same situation as probably we are today with China or some of, other health authorities now entering the world of continuous. And one of the main, I'll say, concern, even though it's not really concern, is just a way from them to understand, because it's a different word compared to batch, is mainly around control strategies. And this resident-time distribution I was talking about earlier, and Robin also explained very well for the dryer, it's new to a lot of people working in the oral solids development. So most of the let's say people in the health authorities, they could not understand how you can control your process and divert, for instance, non-conforming material or tracer material using this RTD. So a lot of questions that were around indeed control strategies.
Thank you, Giustino. Our next question, and I believe, unfortunately, that's all that we have time for, so our last question will be for Dr. Meier. Regarding the continuous drying unit, how can you do feedback control when PAT measures deviations in, for example, moisture content, changed developed speed, but hence as well changing the inlet feed similar to hold up?
Good question. Thank you very much. First thing I need to mention, it's not a belt. It's really a fixed bottom plate. So it might look in the video as a belt because it's steady flow, but it's not a belt dryer or something like this. But nevertheless, really good question for the feedback control of the drying unit, because this drying principle for the first time allows really a feedback control. And it depends, I have to say. The fastest thing to change, where you have the fastest transition function, is really to change the vibration speed. This is done instantaneously, and due to the short residence time, you have around 1 minute perhaps until you see the change of the moisture content in the end, at the end of the dryer. This would be one possibility. The other possibilities are, frankly spoken, all the parameters which can change the drying process or influence the drying process. This is on the one hand the inlet air temperature, also comparably quick inlet air volume, but you can also, as it is conditioned air, change, for example, the air conditioning. For example, change the dew point, increase or decrease the dew point. These are the things which last a little bit longer, and this is up to the control strategy and the control philosophy you have inside your company. But thereby I would only tend to change or to control changes when they are smooth changes. When you have steep and really high frequently changes you observe, then you might have a problem in other processes, like in the twin screw granulation perhaps, because this is usually not a fault of the drying process itself. But what I would never do is change the inlet feed. Let's say change, for example, the twin screw granulator throughput or something like this, because this in the end has affect the throughput of the whole line. And when you do not or did not validate a whole line throughput change, you have in your process not the possibility to really change the throughput of the inlet feed. So it's better to control the nearest control algorithm which is possible, and this in this case would be the vibration speed or different factors which influence the drying process.
Thank you for your answers, and thank you, audience members, for your questions. But unfortunately, that's all the questions we have time for. Before I finish the webinar, though, I would like to ask our presenters, Giustino and Robin, if you have any closing remarks. Giustino, do you have any remarks for our audience?
No, not really. Well, thank you very much, Stephen, for organizing this webinar. I think it's very nice. It was my first webinar, and I really enjoyed it.
Thank you. And Robin, do you have any remarks for our audience?
Yeah, also, I can thank you also. For me, it was the first webinar I gave so far. Was a nice impression, and as I said at the end of my talk, feel free to contact me or us at LB Bohle if you are interested in the technology, want to test or want to visit us. Thank you.
So I would like to take this opportunity to thank our presenters, and once again, that was Giustino di Pretoro, Associate Director of Drug Product Development at Johnson & Johnson, and Dr. Robin Meier, Manager, Scientific Operations at LB Bohle Maschinen, for sharing their knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers back to you. Thank you, everyone. Have a great day.